The next note cards you are going to be filling in will be your light blue note cards, and this will contain information about DNA. DNA is a nucleic acid that stores your genetic information. Since it's a nucleic acid, it means that the monomer that builds DNA is known as a nucleotide. A nucleotide is made up of 3 individual parts. You can see in the diagram there that it is a phosphate, a deoxyribose sugar, and then a base. The base that is in a DNA nucleotide is either going to be an adenine, a thymine, a guanine, or a cytosine. And you can see in that diagram to the right how they match up. A C always goes with a G, and an A always goes with a T. The other nucleic acid that you need to know about is RNA, and you should be able to compare and contrast these 2 nucleic acids. Since they're both nucleic acids, that means they're both made out of the same monomer, which is a nucleotide. So they are both made out of a sugar, a base, and a phosphate, but the sugar is slightly different. So DNA has a sugar called deoxyribose, where RNA has a sugar called ribose. The bases are mostly the same, so they both have an A, a G, and a C. However, DNA has thymine and RNA has uracil. So RNA has a U, not a T. When it comes to the size of the molecules, DNA has 2 strands, while RNA has only 1. DNA has to make a copy of itself before a cell can divide. So we talked about in the cell cycle how in S phase of interphase, a cell makes a copy of its DNA. Well, when DNA replicates itself, it opens up, and each of the original template strands receives a second new strand. So in the diagram here, the original strand is red, and then the new strand would be blue. So for part 3 of the diagram, you can see that of the copied DNA there, each of it has half that's old, half that's new. Therefore, we say DNA replication is semi-conservative. Half of it is old, half is new. So half is conserved, the other half is new. When it comes to actually using DNA to make a protein, there were all these different steps that we learned about for protein synthesis. But just please always keep in mind that DNA is an important molecule. It's kept safe in our nucleus. And the reason why is because it's the directions to make a protein. And so step 1 of protein synthesis is called transcription. And this is happening in the nucleus. And DNA is transcribed into mRNA. Once that happens, mRNA leaves the nucleus. Then the mRNA strand goes to a ribosome, which is the site of protein synthesis. And at the ribosome, the mRNA is read in groups of 3 letters, and it is translated into a protein. So protein synthesis is made up of 2 parts, transcription and translation. And they are in order alphabetically, so that's how you can remember the order of them. During the translation process, tRNA brings in amino acids that are joined to build a polypeptide or a protein. So in this image here, highlighted now, you see the tRNA. They have an amino acid attached to the top of them. And the tRNA will take the anticodon that's on the bottom of them, and they'll match it up perfectly to the corresponding mRNA codon. So these are just groups of 3 bases that are complementary to one another. Notice how you do not see any T's here. They are uracils, not thymine, because this is RNA, mRNA and tRNA. They do not have thymine. And so they're in groups of 3 letters called codons on the mRNA. And this is important because we can use an mRNA codon chart to actually determine the amino acids that will be brought in on the tRNA. So we use the mRNA codon chart to determine the amino acid. So for example, if we look at mRNA codon that is CAU, and we look that up in the table here, the first base is a C, so we're in the second row. The next base is an A, so we go over to the third column. And then that last base is a U. So CAU, it gives us the amino acid histamine, HIS. Okay, so HIS would be our answer there. So please remember how to use this for your state test next week. I'm sure you will see this. The last thing we need to remember for DNA mutations, or when it comes to DNA, is that it can become mutated by different sorts of sunlight, mutagens such as toxins, chemicals, alcohol. Smoking can cause mutations in your DNA, and these are very harmful because DNA codes for proteins. So if you get a mistake in a very important gene, a gene is a part of DNA that codes for something. If you get a mutation in an important gene, then your body can't make, in that one particular cell, it can't make the protein that it needs. And so mutations can be very bad, but they are the source of new traits. So when a mutation occurs in a sex cell, that can get passed on to the next generation, which could cause new traits, which could be good. They are sometimes helpful mutations. All right, this is everything that you need to have on your light blue cards.